we are going to look at the single-cell data signature data views available on lungmap.net. You can explore single-cell data in several ways on our site. Here, we show a new approach to viewing and analyzing gene signatures from cell types and subtypes based on single-cell gene expression data as generated and analyzed by LungMap consortium researchers. Choose single-cell RNA-seq data on the home page. Select View for the third entry in the list of single-cell experiments, a single-cell mouse time course experiment collected with Fluidigm technology. On the experiment page, the left side drop-down contains all genes, specific gene, custom gene list, and signature gene list, all of which can be selected to explore the data. Choose Signature Gene List. This reveals another drop-down on the right side. This right side drop-down contains a list of gene signatures that are named for the specific population of cells that was used to derive the list. There are lists named for cell types, subtypes of those types, and developmental stages from which they were derived. The lists are derived from the analysis of the complete dataset based on statistical comparisons at between cell classes, subclasses, and developmental stages. Let's look at, for example, epithelial overall top 500 genes. This signature gene list contains genes that are highly expressed within the epithelial cell type, but may or may not be variably expressed between the subtypes of that cell type. This list is derived by comparing all epithelial cell types to all non-epithelial cell types within this dataset. Click Go. This brings up a heat map, which includes the genes contained in the epithelial overall top 500 genes signature gene list. Let's explore the heat map in a larger view. Click Larger View, which opens the heat map in a separate browser window. The top left of the heat map view has several tabs, including File, which lets the user import and export datasets, Edit, to change annotations and select specific elements of the heat map, View, which allows the user to zoom in and out and customize the heat map view, Tools, which contains several further analysis methods, and Help, to ask questions of the developers and view the source code. In the top right portion of the heat map view are several icons, including the magnifying glass, which the user can zoom in and out and customize the heat map view, Options, to change the color palette or annotations, Save Image to export the heat map, Filter to select specific cell types or subtypes and/or specific genes, and finally, Color Key to view further details about the color palette. Let's select the magnifying glass icon and then Fit to Window. This enables us to view the complete heat map as opposed to scrolling through different parts of the heat map. The heat map, left to right, has 996 columns, which represent all the cells included in the dataset. Please note, cells other than epithelial are included here, since they are included in the dataset. Top to bottom, 492 rows represent each gene within the selected gene signature list. The genes are ranked from top to bottom based on the average expression, measured by negative log2 of TPM plus 1 across all cells of the epithelial class. You can sort columns by clicking the age, cell type, and/or cell subtype rows above the heat map. A hierarchy of sorting can be performed by holding down the Shift key as you click the row. First, select Cell Type. And next, hold down the Shift key and select Cell Subtype. You will now notice numerical values for the hierarchical sorting. You can further compare signature lists side by side. We will now show you how to do that. Going back to the original dataset view window, select Epithelial Subtype AT1 E18.5 Top 500 Genes and click Go. This gene signature is the top 500 genes from the epithelial subtype AT1 from the mouse stage of E18.5. I know that this is a really strong signature towards lung development. This dataset view can be saved by clicking File and Save Dataset. Let's go ahead and save the dataset to your hard drive. Let's set the file name to AT1 E18.5 and leave the file format GCT version 1.3. This GCT version is in regards to the file format for storing the data. The default is 1.3. Finally, click OK, select the location on your hard drive, and save. Let's look at a third signature gene list, epithelial subtype AT2 E18.5, top 500 genes. AT2 is another major subtype within epithelial cells, and we will also get the genes from the E18.5 stage since I know this is a strong signature gene list. Let's save this dataset to your hard drive as AT2 E18.5. Now, let's move back to the previous window that contains the larger view of the first signature gene list. We will now load the two saved datasets into this heat map to compare these signature gene lists side by side. To load the AT1 E18.5 and AT2 E18.5 datasets into the viewer, click File and Open. Select the AT1 E18.5 file that was downloaded and click Open. You'll see a new window where you decide how the data is loaded and compared to the current dataset. Open Dataset in New tab loads the dataset in a different window, while Append Rows to Current Dataset add rows to the bottom of the current dataset view, and Append Columns to Current Dataset adds columns to the right of the current dataset view. You can also overlay onto Current Dataset, where the color scheme dictates the comparison. Change the setting to Append Rows to Current Dataset, since the comparison we want to conduct is gene lists with genes, which are the rows within the heat map. Therefore, the comparison is most valid by appending the rows. Note that if datasets with different cell types were the subject of the comparison, you would append columns to the current dataset. Once Append Rows is chosen, the Select Fields box will appear. This option allows you to select how the rows or columns will align between the original and added datasets. You can select several different metadata elements. In our case, it is metadata for each column or cell in the heat map, since we selected to append rows to the current dataset. Therefore, let's select Cell Metadata field to align the original dataset and the new dataset. Select Cell for both the current dataset annotation name and new dataset annotation name, and click OK. Let's do the same steps for the AT2 E18.5 file that we downloaded. Now that all the data is loaded, to view the complete comparison of signature lists side by side, click the Fit to Window option. To enhance the heat map view, annotations for the column and/or row can be included or removed. These annotations include the age, cell type, and cell subtype that are shown at the top of the heat map. The annotation for each row currently only contains the gene name. To add annotations for the displayed genes, click the wheel icon to change the settings of the heat map. The Options box appears where the color scheme, annotations, and display can be altered. Click the Annotations tab to add annotations for the genes. The pull-down menu for row and column annotations shows each of the annotation fields that are viewed. For the row annotation, in addition to gene being checked, let's also have the source checked and click Close. Source clearly displays to which dataset the gene belongs. Now, as we mouse over the source of each row, we are shown the signature gene list name where the displayed gene is included. A color scheme can be added to the source annotation to further enhance the visual appeal. To show the color of each source gene, right-click Source and click Show Color, which will color the rows based on which signature list the gene is included. When color is assigned, the source is also displayed in words with a mouse over. You can see there are many genes that are expressed in type 1 and 2 epithelial cells, which is due to an overall epithelial signature, where you have genes that are highly expressed in one of the types, thus being more specific. Therefore, to get a deep understanding of a specific group of cells, we must combine the cell class genes, epithelial overall signature gene list, and the subtype genes, epithelial AT1 and AT2 signature gene lists. Further analysis can be conducted to better understand the differences in type 1 and 2 epithelial cells, including mechanistic pathways. This will be discussed more in our next video. Thank you for your time.